Hello students. In continuation with the previous lecture, we are going to start today with some important terms which are relevant over here. First is gene, unit of inheritance. It is located on the chromosomes. It is also called unit to synthesize a protein. It is responsible for showing the characters in an individual. Another related term which is related with gene is allele. Two alternating forms of a gene are called allele. For example, capital AA, which are both are dominant. These are the two alleles for a gene which shows the flower position axial. Another case, capital A, small a, one is dominant over the other, that is, capital A is dominant over the other, that is, small a. Here, capital A is dominant over the small a. These are the two alleles. These are opposite. One is dominant, one is recessive. Small aa, both are recessive. These are also alleles. The two forms are alleles which are representing one gene for one character. AA is the example for flower position which is axial. Yesterday we did capital TT which was for the height of the plant. Like this we had capital TT, small tt and capital T, small t. The example which we did yesterday. Another example and important term associated with it is homozygous. Both the alleles for a character are same. For example, capital AA, small aa, capital TT, small tt. They are homozygous. Heterozygous, both the alleles for a character are different. Capital A, capital T, small t, capital I, small I. This is the case of the heterozygous. Then there are two more terms. These are the ratios, genotypic ratio and phenotypic ratio. When we do the crosses, we reach up to these ratios. We should understand them. Genotypic, this gives the genetic makeup of the individual of a generation. In the checker box, yesterday we studied 4 in the F1 generation. And in that generation, what is the ratio? We got 1 is to 2 is to 1. That was the genotypic ratio out of the 4. 1 is to 2 is to 1, which is the typical ratio in a Mendelian cross. Then comes the phenotypic ratio. This is the external appearance of individuals of a generation. Again, in that checker box, the ratio 3 is to 1. We will be more familiar with genotypic and phenotypic ratio as and when we do more crosses for the Mendel, like we did yesterday, law of dominance with the help of a cross. Again, I am explaining law of dominance, and here we will be covering the terms which I have dealt right now. Law of dominance is explained by monohybrid cross, means when we take one character at a time. Yesterday we took the character of height. Now I am taking another example, and I am taking the example of the seed coat, yellow versus green. Mind it, yellow is dominant over green. We are using the symbol of the yellow. As yellow is dominant over green, we are not using small gg here. We are using small yy here. Here yellow is dominant over green and I am making the cross over here. Parents yy into small yy. This is the homozygous dominant condition. This is the homozygous recessive condition. When we make a cross between two homozygous, one is pure dominant and one is pure recessive, both are homozygous. In the gametes are like this. In the gamete, we always take single dose. Whatever is there in the parent dose, half we show here. Now in the F1 generation, like this we can make a cross between this and this. Between this and this. Another between this and this. Between this and this. In every case, we get the result capital Y, small y. Means this is the hybrid. This is the heterozygous condition. Apparently, it will be like the yellow. Second step is always selfing of F1 hybrid. Means selfing means taking the same genes both the sides. And I am showing the gametes over here. The parents are here this capital Y small y into capital Y small y. Same because we are doing selfing of the F1 hybrid. F1 hybrid was capital Y small y. We are taking the same parent on both the sides. That is why the same gametes are shown on both the sides. And when we make a cross, we get capital YY over here, capital Y small y over here, capital Y, small y over here, small y and small y over here. Now look at the results. Here we have capital Y, capital Y. Sure, it is yellow. Here it is capital Y, small y. At least there is one capital Y. Means this Y is dominant over this. The result is yellow. Again, capital Y, small y. Minimum one Y is there. Capital Y is there. It means this is dominant over this. The result is yellow. Here is no capital Y. We have small y, and the result is like the recessive parent. It is like green. Now we make the ratios. We have two type of ratios, genotypic ratio and phenotypic ratio, which I just discussed. In the genotypic ratio, genetic constitutions of the individuals is taken, and we get 1 capital YY, 2 capital Y small y, 1 small yy. So the ratio is 1 is to 2 is to 1. In another ratio, that is the phenotypic ratio, we only see the external appearance. Externally, we have 3 yellow and 1 green. So this is the phenotypic ratio. Yesterday we did one example. I asked you to solve more examples. This is another example, and I ask you to solve some more examples so that you may be very confident with all the 7 cases reported by Mendel and in which we can study the law of dominance, which is explained only with the help of monohybrid cross. The other laws will be explained with the dihybrid crosses. But we are not starting second and third law. We are finishing with the first law, that is law of dominance. That will be taken up separately. Here is a new concept. What is test cross? It is also related with the monohybrid cross. Means by using a monohybrid cross, we can use the test cross. First of all, what is a test cross? To find the genotype of an individual, that is, for example, violet color of a flower could be due to capital W small w when capital W is dominant over small w, or it could be due to pure character WW, capital W, both the case. It means if we see a violet flower, it could be due to pure character, it could be due to hybrid character. In both the cases, apparently the flower is violet. But here violet is not indicated with V. By convention it is taken with W. But anyway, the dominant is taken with capital and recessive is taken with the small one. Now, what is the use of test cross? To find out unknown genotype. Why it is unknown? It looks violet, but we don't know the genotype. It could be capital WW pure or it could be capital W small w. How to find out? The rule says cross the unknown individual, the genotype with unknown with double recessive parent. Means small w small w. So we have two cases. Case 1, case 2. In the first case, we take hybrid capital W small w and make a cross with double recessive small w, that is with the white flower. In the second case, we make a cross with capital W and with small w, that is white flower. In both the cases, it looks violet, but we don't know the genotype. So the technique is in the test cross that make a cross with recessive parent, that is pure recessive white flower. In the first case, when we suppose that it is hybrid and we take the gametes like this on one side and the gametes of this on this side, you can take it other way. The result will be same. Here capital W, capital W, small w, small w, small w, small w, small w. If we look at the result, it is violet because capital W is dominant over this. It is violet. Capital W is dominant over this. It is white. It is white. No capital W in these two cases. The result is 50% violet, 50% white. Out of the 4, 50% are showing violet, 50% are showing the white. 2:2. That is why the unknown flower is heterozygous. Heterozygous means capital W, small w. It means if we get this result, 50% like one parent and 50% like the recessive parent. It means the unknown case will be heterozygous. Second case, we suppose with this and we make a cross. These are the gametes shown here and here is the cross. Here we are showing the gametes of this parent capital W, capital W. Here we are showing these gametes over here and we make a cross, capital W, small w, capital W, small w, capital W, small w, capital W, small w. This is dominant over this, violet. Again capital W dominant over this, violet. Capital W dominant over this, violet. Capital W dominant over small w, violet. 100% violet. Unknown flower is homozygous. This is how we can find out the genotype of the unknown individual. So, what is the use of test cross? To know the genotype of the unknown individual. If the individual is pure and we are sure, it means no need of test cross. Suppose it is white. No need of test cross, it is white, it means it is recessive. No need. Had there been a single W, it would be like the dominant parent. Now we are doubtful whether it is this hybrid condition or it is this pure condition. Whether it is this condition or it is this condition. I mean to say, to start with this pure condition or this condition. By knowing the results, we have to simply apply the test cross by making a cross with the double recessive parent. In both the cases, we have to see the results. If the results are like 50-50, 50% like the dominant parent and 50% like the recessive parent, it will be heterozygous. If the results are 100% like the dominant parent, it will be homozygous. So this is the use of the test cross. So today I have finished with Mendel's first law and covered some examples, and here you are also supposed to cover some more examples and do them in written. And then we have discussed the role of test cross. Next lecture will be related with incomplete dominance. As we have covered dominance, the next will be related with incomplete dominance. Because in Mendel's cases, all dominance is obeyed. That is slightly away from Mendel, where we will study incomplete dominance in a very special case. That will be dealt in a separate lecture. Thank you.